Hi everyone, we are Interface Biosciences and we are defining the future of microbiome therapeutics. My name is Hannah Wastick. I'm a bioengineering PhD at Stanford and co-founder. Will Van Churn is also a co-founder and a recently graduated PhD from Stanford's Microbiology and Immunology department. We're led by our PI Justin Sonnenberg, a tenured professor at Stanford with over 20 years of microbiome experience. Now, I'm sure you've all heard of the microbiome, which is that collection of trillions of microbes that live in and on our body, mainly in our intestine. An explosion of research in the past 5 to 10 years has implicated the microbiome in promoting health and disease in nearly every organ system, as well as drug metabolism. Because the microbiome plays a major role in almost half the leading causes of death in the US, there's a huge opportunity to identify drugs or therapeutics by better understanding the microbiome. Unsurprisingly, corporations have invested over $3 billion in microbiome research in the past decade, with worldwide milestone payments eclipsing $5.4 billion. Even without approved drugs, microbiome companies are securing billion-dollar deals with pharmaceutical partners. The current approach used by these first-generation microbiome companies is to give a live microbe as a therapy. These are known as live biotherapeutics, and despite some successes, they have some serious commercial risk. First, they have a limited discovery process due to noisy samples and a small search space. Second, they have serious commercial risks, including high expense to manufacture, unpredictable efficacy based on a person's diet, lifestyle, or genetics, and finally, serve a much smaller patient pool relative to a traditional pharmaceutical. Despite significant research, no live biotherapeutic has been approved by the FDA. These companies are pursuing the wrong approach because they're missing the most important part of the picture. When I showed you this picture before, we used the level of detail that people focus on today. Microbes create some sort of human cell response. What I didn't mention, however, is the most important part, which are the chemicals, otherwise known as metabolites, that microbes produce. These metabolites are small enough to pass through the mucosal layer, enter our systemic circulation, and alter human cell behavior. Interface Bio has the solution to this microbiome therapeutics problem. We focus on metabolites instead of microbes. We're a drug discovery engine that identifies the chemical reasons microbes help people, and we apply those to solving them to many different diseases. I'm gonna let my co-founder talk to you about how we do it.
So here's how we do it. First, we measure the metabolic potential of hundreds of individual microbes. Then we use systems immunology to identify the immune phenotypes induced by each of those metabolites. Finally, we select metabolites that support immune phenotypes and demonstrate in vivo activity. In total, our pipeline allows us to mine the microbiome for novel immunomodulatory chemicals that can be developed as traditional small molecule therapeutics. The database we generate is an unparalleled biochemical resource that only Interface Bio can make. Here, we're showing the most comprehensive strain metabolic map in existence that we've generated. It shows chemical production and consumption for over 200 different strains, each of which is a row, and the columns are individual metabolites, numbering over 2,400. The ability to research and identify candidate metabolites is scalable and benefits from network effects. Our approach takes advantage of predictable pharmaceutical chemistry rather than trying to modify entire microbiomes. We'll show you how this database enables the search for anti-PD-1 immunotherapy enhancing metabolites with some proof of concept data. First, a little background. Immunotherapies are breakthrough treatments that have led to significant advances in cancer therapy success, but they only work in 12% of eligible patients. Recent research has shown that the difference between the responders and non-responders to immunotherapies are their gut microbes. Some metabolite that a microbe is making is improving patient treatment success, but no one has identified it yet, preventing the expansion of the immunotherapy patient pool. We can use the Interface Bio data to identify these metabolites. The heat map we showed before is data on 200 strains, including 4 that are known to support immunotherapies. By comparing strains that support immunotherapies to those that don't, we can identify the potential causal metabolites. The 40 most likely metabolites here are shown in red diamonds. This screening process is unique to Interface Bio and gives us a way to rapidly hone in on the key microbial metabolites responsible for a phenotype that we can further develop. Our plan is to continue to build our database over the next 18 months and expand it in all 3 dimensions. We'll use that data to build out the first clinical indication, which is immunotherapy supporting metabolites. As a drug discovery engine, we continually identify therapeutics within the microbial chemical space across a range of diseases and treatment modalities. We're excited to define the future of microbiome therapeutics, and we're looking for the right partners to help us on this journey. We hope to hear from you soon.